Dear friends, now let us move on to the first question. The question is, the term lead in taxonomic aid used for identification of plants and animals refers to. So, the question is from the first chapter of the first unit of CBSE class 11 or first PUC, and let us look into the options. Paired contrasting characters, option 1, option 2, habitat and distribution of plants, option 3, information for identification of species, option 4, each statement in the key. So, remember what is the key? As the statement says, key is nothing but a taxonomic aid used for identification of plants and animals, where we are going to use various words like manual, fauna, flora, lead, couplet, all this. Let us look into the answer, the statement which is going to be referred to the word lead in the key as a taxonomic aid. Let me come to the first statement, paired contrasting characters. Remember, to identify an organism, this is very important. We are going to look into the statements of opposite nature, what we call contrasting characters. For example, if we come across a snake, to identify whether the snake is venomous or non-venomous, the first thing that we look is the tail. Tail, blunt end, pointed end. Blunt, non-poisonous, pointed, poisonous or non-poisonous. We call such statements of opposite nature as couplet. So, here paired contrasting characters relate to what do we call as couplet. So, that is not our answer. Let us come to the statement 2, habitat and distribution of plants. Whenever we talk about plants and animals, the habitat is the place where they are going to be found, and in case of plants we use the word flora, for animals we use the word fauna. So, statement 2 relates to flora. Let us come to the third statement, information for identification of species. So, where do we get it? In the key? Then remember, it is in manuals. I hope all of you remember, in the practicals you study the information what is given in the laboratory manuals. So, here information for identification of species relates to manuals. Finally, we come to the fourth statement. Each statement in the key is called as lead because it leads to the next aspect. So, the answer is option 4, that is each statement in the key is called as lead. Now, we come to the next question, which criteria the R.H. Whittaker, underline, remember this, R.H. Whittaker's 5 kingdom system classification, I call it 5 kingdom classification, does not include. Remember, we have studied R.H. Whittaker's classification, and also in the book there is a mention of the 2 kingdom system classification of Carolus Linnaeus or Carl von Linné, where the 2 kingdoms are Plantae and Animalia. And at the same time, I want you to know the 3 kingdom system and 4 kingdom system classification also. The 3 kingdom system classification, I hope all of you remember, was given by Ernst Haeckel, where he is going to add 1 kingdom to Plantae and Animalia, that is Protista, and co-plants 4 kingdoms classification includes Monera, where all prokaryotic organisms were removed from the Protista and placed in the kingdom called Monera. Protista also included fungi, which is called as kingdom Mycota, and that separation was done in the 5 kingdom system classification of R.H. Whittaker. R.H. Whittaker has used 5 different criteria or bases to classify the organisms into 5 kingdoms, and here let us look into it. Thallus organization, yes, he used as a criteria, a basis. Mode of nutrition, yes. Phylogeny, that is evolution. In addition to this, he also used cell structure and organization and reproduction. Please remember, these are the 5 criteria used by R.H. Whittaker for his 5 kingdom system classification, that is thallus organization, mode of nutrition, phylogeny, cell structure and organization, and reproduction. So, habitat was not used, and thereby here does not include. So, the right option is habitat. Now, we come to the question, diatoms and desmites are regarded as planktons. Note down, planktons, because they, options, option 1, are similar to plants, are minute and free floating, are minute and active swimming. Option 4, have 2 thin overlapping cell wall. Dear friends, most of the students get confused when they come across the options, because many of the options go with diatoms and desmites. Remember, diatoms and desmites are now placed in kingdom Protista. They are chrysophytes. I hope all of you remember, Protista include chrysophytes, dinoflagellates, for example, goniloques, red tides, I hope all of you remember, Noctiluca not given in the text, euglenoids, slime molds and protozoans. And diatoms and desmites are called microjewels. Earlier they used to be called as microjewels of plant kingdom. The reason is, earlier they were part of the plant kingdom, now Protista being unicellular. They are algae. For example, desmites are called as golden algae. But here we are not looking into that option which go with the diatoms and desmites. We are looking for an option which is going with why they are referred to as planktons. When you talk about planktons, please remember, free floating organisms, minute free floating organisms are called as planktons. Minute free swimming organisms are called as nectons. And here when you look into it, planktons, so naturally it go with the option are minute free floating organisms. So option 2 is correct. Of course, they are similar to the plants because algae. Remember, algae is that which is going to be representing as monerans, for example, blue-green algae, cyanophycean members. And diatoms and desmites, Protistan kingdom. Green alga, brown alga, red alga, they represent plant kingdom. Second one, we know, minute actively swimming organisms, these are called as nectons, free swimming. They can move from place to place by their activity, they can get displaced. Last one have two thin overlapping cell wall. That is one of the feature of diatoms. Diatom cell wall, cellulosic wall is made up of two thin overlapping shells, and this cellulosic wall is embedded with silica that makes it indestructible. That is the reason why we have diatomous earth. So anyhow, we know the option 2 is the answer. At the same time, we know what others also refers to. Please remember, minute active swimming forms are called as nectons. Planktons of plant origin are called as phytoplanktons. The planktons of animal origin are called as zooplanktons. Now we come to the next question. The gelatinous coating material on the cellulosic wall of brown alga is. Question is quite simple. Let us come to the option. First option, laminarin. I hope all of you know laminarin is reserve food material of Phaeophycean members, that is brown algae. Mannitol is also reserve food material. On the cellulosic wall, the gelatinous covering what we find is algin. Thereby, it must be the answer. Let us look into the option 4 also. Floridian starch. Remember, Floridian starch is a reserve food material in red algae. It has structural similarity with amylopectins and glycogen. So thereby, first option 1, option 2, option 4 relates to reserve food material. First 2 relates to reserve food material of brown algae, the fourth one relates to reserve food material of red algae. So answer is algin. It is very important from examination point. So remember the answer. Correct option is 3. Now we come to the next question. Which phylum is characterized by features like radial symmetry and tissue grade of organization with exclusive marine animals? Options are option 1 Porifera, option 2 Coelenterata, option 3 Echinodermata, option 4 Ctenophora. Let us look into the questions and one by one we discuss this. Which phylum is characterized by features like radial symmetry? The moment we come to radial symmetry, we know that radial symmetry is exhibited by all the 4 phylum members: Porifera, Coelenterata, Echinodermata and Ctenophora. But the difference is, in Echinodermata, we are going to find organ grade of organization. Please remember, in echinoderms, adults exhibit pentaradial symmetry, whereas larvae is bilaterally symmetrical. Larvae is more advanced compared to the adult. Thereby, we call the type of metamorphosis what we see in Echinodermata as retrogressive metamorphosis. So thereby, Echinodermata can't be an answer here, because here it is mentioned tissue grade of organization. We are going to eliminate this. So in Echinodermata, we are going to find organ grade of organization. Radial symmetry, tissue grade. Again, when you talk about Porifera, sponges, and here in sponges, this is also not an answer because they exhibit cellular grade of body organization. So either answer is coelentrates or ctenophores. Now we have come to the option 2, option 4. The third says exclusively marine animals. They are found only in marine, that is sea water, not freshwater forms. And when you talk about exclusive marine forms, remember echinoderms are exclusive marine forms, ctenophores are exclusive marine forms, but these are organ grade. So answer must be ctenophores. What about coelentrata? Coelentrata, we have freshwater forms as well as marine forms. The classical example for freshwater form in coelentrates, where we have tissue grade of organization, is hydra. I hope all of you remember. Example, hydra. Hydra is a freshwater form. So thereby option 4 is the right answer. In addition to this, do we have to know something else about ctenophores? Then remember, in ctenophores we are going to find only sexual reproduction. There is no asexual reproduction. I hope in case of hydra we are going to find budding. Hydra exhibit high power of regeneration. In case of coelentrates we are going to find alternation of generation. In coelentrates we have polyp-like, medusa-like forms. And in coelentrates, the alternation of generation from asexual form to sexual form and vice versa is called as metagenesis. Remember that also. Now, when you talk about ctenophores, only they exhibit sexual reproduction. They are commonly called as comb jellies, sea walnuts. Ctenoplana, plurobranchia are examples that you have to remember when we talk about ctenophores. With this, now we move on to the next question. The question is, an organism shows spindle-shaped body was caught in the fisherman net. So a statement is there. It had circular mouth, important, scaleless body, and was devoid of paired fins. Which among the following is the most likely organism? Let us look into the options. It is a hemichordate, a cyclostome, a cartilaginous fish, a bony fish. So when we look, spindle-shaped body, it is fish-like, but is it a fish? Circular mouth, scaleless body. Of course, when you talk about catfishes, we are going to come across scaleless body, but mouth is not circular. So thereby it can't be a bony fish. Devoid of paired fins, all fishes, whether cartilaginous or bony fishes, they are going to have paired fins, like pectoral fins, pelvic fins. And thereby bony fishes and cartilaginous fishes are not our options. They are not the right answers. So now we look for Hemichordata and Cyclostomata. Hemichordata is an invertebrate phyla, Hemichordata. You remember, you have studied Belonoglossus, where the body is elongated worm-like. So thereby even this is not an answer. No fins, of course, no scales. And when you talk about cyclostomes, cyclostomes are placed in the group Agnatha. I hope all of you remember, chordata include Agnatha and Gnatostomata. Jawless vertebrates. Gnatostomes are jawed vertebrates. When you talk about Agnathans, It includes a class called cyclostomata, circular mouth, cyclostome. The organisms which have circular mouth because jaws are absent. Two examples you have studied. One include lamprey, the other is myxin. Lamprey is called petromyzon. Myxin is a hagfish. So, in both the cases no scales, no paired fins, unpaired fins are present. Remember, lamprey is also sanguivorous in habit, that is the one which feed on the blood of the host. So, thereby as the circular mouth, scaleless body, devoid of paired fins, all these three go with this, the right option is cyclostomata. These three are not the answer for us. The question is, select the wrong statement. Let us come to options. Gourd cells of grasses are dumbbell shaped. I hope all of you know grass is a monocot. When you talk about stomata, stomata are the openings through which transpiration occurs. When we talk about transpiration, the main type of transpiration is stomatal transpiration. It is also called foliar transpiration because leaves are the main organ where stomata are present and they are going to lose the water in vapor form through stomatal opening. The stomatal pore stoma is bound by gourd cells. In dicots, the gourd cells are bean shaped or kidney shaped, whereas in monocots it is dumbbell shaped. Since grass is a monocot, dumbbell shaped, so option statement is correct and we have to look for wrong statement. So, the option 1 is not our answer. Now, we come to the option 2. Trichomes check transpiration. What are trichomes? This question is related to plant anatomy, structural organization in plants. Multicellular epidermal hairs are called as trichomes. Unicellular extensions of epiblema, rhizodermis or piliferous layer is root hairs. When you talk about trichomes, they are multicellular epidermal hairs, they may be branched or unbranched and they are going to have a coating of cuticle. So, thereby they are not going to help in transpiration, they prevent transpiration, they check the transpiration. So, option 2 is a right statement and we are looking for wrong statement, thereby it is not our answer. Now, we come to the statement 3. Option 3 says only parenchyma constitute ground tissue. Remember, parenchyma is a simple permanent storage tissue. It is quite abundant in the plant body. When you talk about tissue system, in your text you have studied, there are 3 tissue systems: epidermal tissue system, ground tissue system, vascular tissue system. So, other than epidermis and vascular tissue, which is a compound permanent tissue, rest of them are going to represent the ground tissue. That means it includes all the 3 simple permanent tissues, namely parenchyma, collenchyma and sclerenchyma. So, thereby the option 3 which says only parenchyma constitute ground tissue is a wrong statement. So, the option 3 must be the answer for us. Let us come to option 4 also. Phloem parenchyma is absent in most monocots. When you talk about monocots, especially the stem, we are going to see irregularly scattered vascular bundles, what we call as atactostele condition, where we are going to have the phloem and in the phloem the parenchyma is going to be absent. That is a characteristic feature. Similarly, when you talk about the xylem, only 2 metaxylem vessels and few 1 or 2 or few protoxylem vessels with fluid filled cavity is going to be present. So, since here phloem parenchyma is absent in most monocots, that is also a right statement, thereby this is not our answer. In addition to this, I want you to remember, in case of monocot stem, the pericycle is going to wrap each vascular bundle and it is in the form of bundle sheath, whereas in dicot stem it is in the form of bundle cap. The bundle cap, the bundle sheath of the stem is always represented by sclerenchyma tissue.